Gonna stand over here. All right, I gotta press this button. Got it. All right, thanks for having me today, everybody. As Mel said, my name is Matt Vaughn. I'm the Chief Scientist at Lake Champlain Basin Program. I'm really happy to be with you. I don't think I've been asked to give an hour-long lecture on Lake Science 101. It's a very broad topic, and I was excited at first, and I was like, what am I actually going to say? There's so much to say here. But the good thing is I'm sure you all love lakes. So who here lives on or near probably Lake Carmi? Most of you, almost all of you. Wow. Yeah. So I'm sure you love Lake Carmi. I'm sure you care deeply about Lake Carmi or other lakes. I work for the Lake Champlain Basin Program. I'll talk about more about what we do. Our focus, of course, is Lake Champlain, but that includes the entire watershed that drains the lake. So I'll talk about that as well, which of course includes Lake Carmi. So I care deeply as well. We're all here to learn. And as Mel said, we have a small group here. I'm happy to have this be conversational and feel free to ask questions as we go. And I think we have a question and answer discussion period after the talk as well. So if you have kind of a bigger issue or question to dive into, you can hold it for the end as well. But I wanted to start, I thought it'd be fun to see, these are my two kids, by the way, enjoying Lake Champlain. I wanted to know if anyone could try to define what a lake is. Body of water. Body of water. Okay, but I'll just put one big puddle, body of water. Or I think my next one is similar. A hole in the ground that's full of water. Right. And these, there's a lot of correct definitions. But anyone else? Yeah.
It doesn't have vegetation and totally covering it at any time.
Okay, wow, that's a great definition. Yeah. Not totally covered by she said it doesn't have vegetation totally covering it at any time of the year. That might be more like a wetlands instead of a lake or a pond. Okay. Yeah.
It's a complete ecosystem.
Complete ecosystem. That's a cool one. All right. Yeah.
Surrounded by land on all sides.
Surrounded by land on all sides. I like that too. All right.
The sun can't hit the bottommost point.
The sun can't hit the bottommost point. I never thought about that. I like that one too. Okay. All right, I have a few more for you. A habitat for wildlife, fish and birds, right? A place to cool off on a hot day, right? We all like that. A place to explore, ideally it would be, right? A source of drinking water. And so for this talk, this is kind of, in my work, those are all kind of with my kids or when I'm a normal person. But in my work, I kind of wrote this down just for this presentation, a freshwater ecosystem. So we have that with plants, animals, and bacteria, where energy and nutrients are transformed and transported. So sciencey words, right? Through physical and chemical processes. That's not all a lake is, but it's part of what it is, right? And so I wanted to kind of touch on some of these things. Hopefully it'll be interesting to you. And I was assuming coming into this that many of you are engaged with Lake Champlain, or probably most likely Lake Carmi, especially, and have a basic knowledge of lakes and our water quality challenges. You probably saw it this morning out your window. So I'm not going to focus entirely on cyanobacteria or nutrient loading, which is you've probably heard a lot about. But I'm going to talk about a bit of how lakes work from from a process standpoint. And then I think that's going to hopefully inform our understanding of these issues that we're facing. And toward the end, I'm going to talk more specifically about cyanobacteria and, the case of Lake Carmi specifically. And hopefully we'll be able to draw from what we learned earlier and kind of touch on the things we're facing that you probably are very aware of. So I want to start with the fun word, limnology. You all heard this one before? Few of you, the study of lakes. So congratulations on your first day of being a limnologist. I like to tell my kids all these sciencey words, like things that I am: a limnologist, a hydrologist, a study of water, a fluvial geomorphologist, someone who studies how rivers change. Those are all kind of fancy words for just studying water. And as I said, we all care deeply about these things. They're critical for drinking water, for our ecosystem, and we want to keep them in good shape. So that's why we're here. So before we talk about lakes themselves, I wanted to look uphill to kind of watershed science, because I think it's really important to look at the landscape setting before kind of diving into the lake itself. So this is another thing I kind of wrote down thinking about Lake Science 101. Lakes are at the bottom of something and in the middle of something else. If you think about it on the landscape. So at the bottom is the ocean, right? Wherever you are, if you're at the bottom of, unless you're talking about Death Valley, but in general, water moving downstream is going to end up at the ocean. If you're in a lake, a freshwater lake, you're at the bottom of its watershed. We'll talk about that here. Watershed is all the land that drains to a point. So if you think about a lake's watershed, it's all the land that drains to that lake. And you can see this is I like this diagram. I just found it online from Hudson River folks. You can see that you probably all have heard about watershed divides along maybe the ridge of a mountain line or a mountain chain. But watershed divides can also be just, along the top of a rolling hill. It doesn't have to be a very, drastic part of the landscape. I lived on a watershed divide in Burlington for many years between what drains to the Winooski River and what drains to the lake directly. It's kind of a minor watershed divide. It was still within, of course, the Lake Champlain Basin, but it's a smaller divide between one big watershed and a small one. So watershed divides happen all over the place. They can determine whether water is flowing to a big river, to a small river, directly to a lake, to the Pike River or the Missisquoi River, for example. And watersheds determine the inputs of course, water, but also sediment, nutrients, pollutants, everything that goes along with water as it's moving downstream. I think that's, and then of course, so watersheds can go to a point. So any point in the landscape has its own watershed. Lakes, are something we care about. So when we're managing lakes, we're thinking about lake science, we're looking uphill and what drains to this lake. So I think my next one, right, is a couple maps here. So sorry if this is kind of hard to see or too small, but on the left we have a map of the Lake Carmi watershed. So you may have seen this before. And then on the right we have a map of the Lake Champlain Basin or Lake Champlain watershed. So I just wanted to point out a few things on these two maps. So Lake Carmi watershed is, I think it's 7,700 acres, which is around 12 square miles. I just looked these numbers up. And then the lake itself, I think, is around 2 square miles. So an important thing to think about is how big is the watershed compared to the water body itself. So in this case, if you subtract out the area of the lake from the watershed, that's the amount of land you have, right, draining to the lake. And then you divide that by the amount of surface area of the lake, you get about 4.5. So there's 4.5 times as much land as there is water. Okay. Contrast that with Lake Champlain. We have an over 8,000 square mile watershed, and the surface of Lake Champlain is about 435 square miles. So that ratio is 18 to 1. So on the left, Lake Carmi, 4.5 to 1. On the right, Lake Champlain, 18 to 1. If you look at other major lakes in our region or in our country, the Great Lakes, this is about 1.5 to 1. Some of them are about 3 or 4 to 1. So this really determines how much water, sediment, and nutrients are making their way to a water body, and has big implications for how to manage that. Basically, what is the source of our potential pollution? Water and sediment. And of course, Lake Carmi is positioned within the Lake Champlain Basin. So it's not, it should be a little blue dot there, but there's that. All right, now I want to talk about from a watershed perspective, different sources of pollution. And we call them point source and non-point sources of pollution. Has anyone heard those before? A few of you? Okay. So it's pretty easy to remember because point source is something you can point to. Things like wastewater treatment facilities, combined sewer overflows, industrial sources or factories, large animal operations, farming operations. These are things that you can point to, and they're more or less, they're easier relatively to regulate. And these are things that the Clean Water Act has made enormous strides on since the 1970s. So point sources of pollution, we can point to them, we can manage them, we can regulate them. And we have technologies to reduce pollution from point sources that are really effective. Non-point sources of pollution come from the landscape. They're more diffuse. You cannot point directly at them. So I'm gonna just scroll through a few pictures of our landscape here, you know, in the region of Lake Champlain Basin. This, of course, is a forest. It is relatively, you know, not as great a source of pollution as other landscapes, but there are potential sources of pollution here from this, again, non-point sources of pollution. Forest roads, logging roads can erode and take sediment downstream along with nutrients and possible other pollutants due to development in forested areas. Forests, you know, have a long history of deforestation, reforestation, and their you know, plant and tree diversity has been affected by that. They get attacked by a fly. Of course, agricultural landscapes have their own sources of pollution. We'll talk more about this later, but phosphorus is brought onto farms to fertilize crops and to feed animals, and an excess of these nutrients like nitrogen and phosphorus run off to waterways and make their way to Lake Carmi, Lake Champlain, etc. Again, land use here, this is a mix of forest and agricultural landscapes is typical of our region. You can see the rivers kind of winding through this mosaic or patchwork of land uses. You can imagine how non-point sources of pollution can come from each of these in a different way. And finally, of course, Burlington is a different type of landscape. You have developed lands, you have, we call impervious surfaces, you know, areas where it rains on the ground and it cannot infiltrate into the soil. It runs off immediately and it brings pollutants and dust and nutrients along with it. So, you know, developed areas have their own challenges and own set of technologies and solutions to try to reduce pollution from areas like our cities. Then I also wanted to, I'm going to get back into this soon, but the atmosphere, the air is another source of nutrients, energy for lakes. So it's almost like a part of the watershed. It's not really, because watershed is land, but it's something that, you know, maybe you haven't really thought of before or haven't considered. But, you know, our atmosphere, of course, has oxygen. So that's going to fuel respiration in the lake. So that's animals breathing or plants using their energy. It has carbon dioxide, you know, goes back and forth between the water and the air. So plants, and we'll talk more about primary producers, can photosynthesize and make their energy from sunlight. And then phosphorus also, nitrogen and phosphorus can be deposited from the atmosphere, from rain, from dust, right onto the water surface, and that's another source of these nutrients. So that's something that I just wanted to bring up, maybe something you hadn't known before. All right, now I want to talk about rivers, which are, of course, rivers are us. They connect the watershed to the lake, right? It's a point of connection. So whatever is coming off the watershed, it's going to collect into streams. Those are going to flow downstream into bigger rivers, and that's going to connect all of these sources of water, sediment, nutrients to a lake like Carmi or Lake Champlain. So this is a great image. This is not from July. This is from Tropical Storm Irene in 2011, but you can really see what's coming downstream into Lake Champlain. You can see that sediment plume there, and you can see the connection between the watershed and Lake Champlain. So rivers, again, kind of thinking about what's uphill from the lake, really connect the landscape to the lake, and will have really important impacts or sometimes benefits, depending on the situation, to water quality in the lake and to the ecosystem that's, you know, thriving, hopefully, in the lake we're talking about. All right, so thinking about rivers for a few minutes, I wanted to talk a little bit about what we know about rivers and how we learn about rivers. And there's a really key question I thought maybe you hadn't heard of before, which is, how do we know how much water is moving downstream in a river? And this is kind of a fundamental question that a lot of people might take for granted, but it's something that, you know, is developed over decades, and scientists figure out how to measure something we call discharge. It's a volume of water per unit time. So it's something that we measure in cubic feet per second is the common one, or cubic meters per second, how much water is moving downstream. And as you can imagine, this is going to be critical for limnologists like yourself to determine how much phosphorus is moving downstream, how much nitrogen, all these things that are critical for lakes, but also can be in excess or a form of pollution. So to measure how much water is moving downstream in the river, we basically break it up into little chunks. We have, you can see these little rectangles here. And people, generally the US Geological Survey does this in the US. And they do that right here in Lake Champlain Basin. They go out and they measure water velocity or water speed moving downstream for every little chunk of river. And then they multiply that by the area of that rectangle. Okay, and through this map and through this field work, they can say, when I was out there on, what's today, September 5th, I took all these measurements and I added them all up and I get 2,000 cubic feet per second. Okay, and we'll talk about how we use that number in a few minutes, but so that's what we call discharge, the amount of water moving downstream per an amount of time. And then they put these things called gauging stations in our kind of most important rivers around a region around the country. And here's a few diagrams of them, or, one's an image on the bottom right here. This one's in Alaska. And the top two show examples of how rivers are gauged. So a gauge is something that measures the depth of water very frequently, about every 15 minutes. Okay, so people go out and they measure discharge, like I explained, taking water speed measurements and adding them all up. And they say, okay, the river was at 3 feet stage today, and they write that down. And they come back during a flood and they do the same thing. River's now at 8 feet, and here's the discharge that I got. And they do that many, many times over many different water flow conditions. And then they come up with a graph like you see in the bottom left here. So you can see all those little dots represent a time when someone went out and stood in the river or used something from a bridge. This river flows really high. And they measured the amount of water moving downstream, and they measured how deep the water is, and they graph it. And then they put a line through that to try to come up with a relationship. And that way we can know, based on how deep the water is every 15 minutes, about how much water is moving downstream. And so we have stations like that all over the country. There's thousands of them. We have probably 30 or 40 of them in the Lake Champlain Basin. About 22 are right near the lake, gauging how much water is moving right into Lake Champlain. And from this, we have real-time knowledge of how much water is moving downstream. This one is at the Missisquoi River in Swanton. You can see a graph of discharge, so that's cubic feet per second on the vertical axis there, and dates in late August going horizontally. So you can see the amount of water moving up and down. We're ranging here from 500 to 2,000 cubic feet per second. Okay. Now that we have the amount of water moving downstream, we can combine that with some water chemistry. So if I were to go to the Missisquoi River and take a bottle of water and bring it to a lab, I could know how much phosphorus is in there, how much nitrogen, but it's only what's in my little bottle of water, right? And what we do is we measure something called a concentration. So that is the mass per volume. So how much, usually for phosphorus, it might be, 30 micrograms per liter. So that's a mass per liter, which is volume. And then if we multiply the concentration by the amount of water moving downstream, we get something called the load. So that's the mass of, say, phosphorus moving downstream to the lake over amount of time. So maybe second or day or year. That way we can estimate how much of something we care about is entering a lake by combining these ideas. All right, so that was a lot about watersheds and rivers. And now I want to kind of look downstream back to lakes. And I want to talk about water temperature and water density, because this is a really critical concept for all of our lakes. And water has a really cool, really unique property. It has to do with its density. So water at its freezing point, which is 0 degrees Celsius or 32 Fahrenheit, as I'm sure you know, is actually less dense than warmer water. And that's why if you drop an ice cube in your drink, it will float to the top. But if you think about this for a few moments, that's really unusual. Most things kind of condense when they get colder and freeze. Most liquids would act like that. But water, because of its molecular structure, when it gets to its freezing point, it locks up into something called a crystal lattice. and that makes it less dense and allows it to float. And that's why you can see me standing on the frozen surface of Lake Champlain. If water got more dense when it froze, of course, it would sink to the bottom. And we would never be able to stand on top of a lake. Your ice cubes would sink to the bottom. That's not what happens. So we have this curve here where here you're all the way to the left is water at its freezing point. Say, think of an ice cube. And that's the line is lower, so it's less dense. So it's floating on more dense water. And then at 4 degrees Celsius here, that's the point at which water is its densest. That's about 39 degrees Fahrenheit. Okay, so that if water is at that temperature, it's going to sink to the bottom, which is a really important idea. And then as you get warmer from there, it continues to get less dense and will float again. So this kind of a quirky part about how water works with temperature is how we get stratification in the summer. So I'm going to talk about that now. So lakes. Yeah, so lakes do some crazy things throughout the course of the year, and this is there's different types of processes depending on how deep a lake is. So for a deep lake, we call it dimictic, meaning that it mixes twice a year. Di to mictic, mixing. So in the summertime, if it has enough depth, the water at the top of the lake will warm. Okay, and that causes it to be less dense and kind of float on the surface, where the cooler water is still sinking, that's more dense down at the bottom. The place where it changes rapidly is called the thermocline. Okay, that's where there's kind of a boundary between the two layers. And this is really important because, and this is a stratified lake, it has 2 different layers. And lakes that are stratified, the 2 layers don't really mix. Okay, and that's an important concept because that means whatever's living at the top of the lake cannot get what's at the bottom, and there's no interaction, no meaningful interaction between the 2 layers. And then as things begin to cool and you get these windy fall days, that top layer at the surface cools down and begins to sink. And in the fall, we get what's called turnover. So the lake mixes with those bottom layers, and whatever is on the bottom, if there were nutrients or kind of lower oxygen water, that's going to get mixed up with what was at the top. And then in the winter, as things begin to freeze or get close to freezing, that the cold water at the top freezes, and then all that 4 degrees Celsius water kind of hangs out the bottom. That's what's most dense. And then in the springtime, when the ice melts and that top layer starts to warm again, that will sink and then mix again. So we get spring turnover, until we're back to summer, when the water will warm to the point where it floats at the top, sinks, the colder water sinks to the bottom, and it stratifies once again. So that's the annual cycle of, again, a dimictic lake. A polymictic lake is one that is not so deep that it can stratify like this, and that goes through periodic moments of stratification, where if you have hot days, not a lot of wind, it can start to stratify, but then, a windy day or a cool day can just mix it, at basically at any time. So it's much different from a dimictic lake, and some lakes are kind of in between the 2. And I'm going to come back to this in the specific case for Lake Carmi. But I wanted to show you kind of an interesting graph of, this is the main part of Lake Champlain near Valcourt Island. And you're seeing the depth below water surface on the vertical axis here. So the water surface is at 0 at the top, and then all the way down is 150 feet deep. And the color is showing the temperature of the water. So you can see there's a sharp, difference between this warmer layer and the cooler, the purple colors means cooler temperatures down below. So this would be the top layer is called the epilimnion, that warmer layer up above, and that is not mixing in a meaningful way with the hypolimnion down below, these purple colors. And so we can see this is kind of over throughout a few weeks in August, what the water temperature was doing. You can see that the thermocline, the place where it changes between the 2 layers, can kind of go up and down a bit, but there's pretty much solid 2 layers throughout this summer season. Yes.
how would, this would be the natural aspect. So could you speak to how the aerators would be changing this natural aspect of stratification and how it might be giving nutrients?
I'm so glad you asked, because it's my next slide.
Okay.
You set me up perfectly. So I'm gonna get there through a bit of a new concept or a term called the Schmidt Stability Index, it's called. So this is the amount of energy it takes to mix a lake or a piece of lake, really. So going back, I'll kind of flip between these two. So in the main part of the lake, this is a big, deep water body, Lake Champlain, right? It's a deep part of the lake, over 150 feet. You can imagine if you have really cool, dense water down below, or think about it as like oil in water, right? Oil is going to be kind of floating on top. Water is not really oil like this, but you know, floating water on top, dense, heavy water on the bottom. It's going to take a lot of energy. If you're losing the glass, you really have to shake that up a lot to get it to mix. If the temperature was not so different, this is what happens in the fall, right? When it starts to cool. If the temperature of the water on the top is similar to the water on the bottom, you give it a little shake and it mixes readily. Right, and this is kind of what it's like for a polymictic lake, one that doesn't have the depth to have those really strongly different layers. So Missisquoi Bay in Lake Champlain is like this. It's not deep enough to have a large difference between the top and the bottom. It can start to stratify over a week or so, but then a windy day mixes it all up, a little shake mixes it all up. So Lake Carmi is also like that. It has a bit more depth than say Missisquoi Bay in that it can stratify for periods at a time, weeks at a time, even depending on the weather conditions. But a really windy day can mix it up. And what's your name, sir? Paul. So what Paul was mentioning was a secondary effect of the Lake Carmi aeration system. So I'll talk a bit more about the goals of the aeration system and what is the primary purpose. But a secondary effect of this that we discovered through monitoring was that the energy it took to shake it up decreased as a result of the aeration. So basically, where more naturally without aeration, we would have had a stronger difference between the top layer and the lower layer. It still could be mixed. Of course, it's polymictic. It could be mixed. But because aeration was moving things around, the difference was not as great between the top and the bottom. So less of a breeze was able to mix it up, right? And we'll talk more about how phosphorus moves around a lake in a few minutes, but that probably led to phosphorus that was coming up from the bottom sediments of Lake Carmi to be more readily mixed to the surface where phytoplankton like cyanobacteria could use them to grow. So maybe I'm getting ahead of myself here, but I just wanted to point again to how this works with water density and Lake Carmi. This is again the stability index. So the higher number means it takes more energy to mix up. You got to shake the glass, shake the bottle, and the lower number means just a little swirl will mix the glass or mix the lake. So in 2018, there was no aeration system. You're seeing higher numbers, greater amount of energy to shake up Lake Carmi. And then 2020, I believe it was the first year of aeration. It took less energy to shake up Lake Carmi. So that was again a secondary effect of aeration. Going back to again that graph of water density, it's all about differences in density.
Can you explain that graph again?
Definitely. So yeah, I should have explained it more clearly. So the vertical axis going up and down is the density of water approaching. So we're talking about kilograms per cubic meter. It's just the amount of mass per volume. Okay, so higher number means for the same amount of volume, it's heavier, it's denser. Lower number means it's lighter, so it's more likely to float. The horizontal axis is the temperature of water. So all the way to the left is 0, the freezing point, and all the way to the right is 10 degrees Celsius. So it's showing that the freezing point is less dense than 4 degrees Celsius or 39 degrees Fahrenheit.
And you're finding the graph that had two curves on it, two lines on
Yes.
it. That one.
This one, yeah, so this one again, this is this idea of the stability index. So how much energy does it take to shake up a lake and make it mix? So think about it again as a glass of oil and water. That would take a lot of energy to mix it up. You got to really shake it.
But which line is what?
Sure. The red line is how much energy it took to fully mix Lake Carmi in 2018. And the blue line is that same idea, but for 2020. And it's showing the difference because in 2020, there was the aeration system installed,
Thank you.
and that had an effect on the stability of Lake Carmi. Okay. All right, now we're going to switch. I was told to talk about biology, so we're going to talk about biology a little bit. I think the name of my talk was changed a few times. They were throwing words in there from time to time, so it became biology 101 at some point. So I feel obligated to talk about biology a little bit. So, well, you can't even read the words on here. That's too bad. I have more words that are in gray, but anyway, we want to talk about primary producers of lakes. These are the bottom of the food chain. Primary producers just mean they create energy from sunlight. They photosynthesize, right? You remember this from school. So there's generally kind of three buckets of primary producers that are taking energy from the sun and creating food for the base of the food web. There's periphyton. These are things that stick to rocks and sediment, and they grow, at the bottom of rivers and lakes. Periphyton, fun word. Phytoplankton, also primary producers, and these are things that are generally free floating in the water column. So this includes things like diatoms. Diatoms are a type of algae that are super cool. They incorporate silica into their cellular walls, and they make these beautiful shapes under a microscope. And they include, so phytoplankton also includes cyanobacteria, which you're all familiar with, I'm sure. They also include green algae. These are things that are free floating in the water column, photosynthesizing. And finally, macrophytes, which are plants, aquatic plants. Fancy word for bigger plants. Green algae, also a plant, but it's microscopic for the most part. And macrophytes, just macro, bigger plants, photosynthesizing. So let's talk a bit about cyanobacteria. So I just mentioned cyanobacteria is included in that bucket of phytoplankton. Yeah, I'm assuming you all are familiar with what cyanobacteria is, probably too familiar, more than you want to be. I often have to, start from a place of, what is it? What does it look like? I don't think I have to start there with this group, unfortunately. But I want to put it in context a bit. So cyanobacteria, maybe you've heard, is native to almost all ecosystems on Earth. They're incredibly ubiquitous in the soil, on the surface of everything. At basically any surface you look at, there's going to be cyanobacteria in root systems, in aquatic systems like lakes. They are part of the phytoplankton community. So throughout the season, naturally, in a lake that is not challenged with excess nutrients, you might have diatoms kind of blooming or being more a bigger part of the population in the spring. You might have green algae coming through in the later spring and summer, and then you might have cyanobacteria be more populous in the later part of the season. That might be a natural cycle. What we see here with, throughout the world, it's not a problem that's unique to Lake Champlain, not unique to Lake Carmi or even the Northeast. Throughout the world, we're seeing an excess of nutrients in our freshwater bodies, and that fuels, increased primary productivity by all of these things that we talked about: periphytons, phytoplankton, and the macrophytes. Cyanobacteria just have an unfair advantage for multiple reasons. One of the biggest things is that among the phytoplankton community, cyanobacteria are the only thing that have learned how to float. Other types of phytoplankton are just kind of free floating around the water column. They have no way to move themselves, and they don't have a brain. Cyanobacteria, they're not thinking I need to float now, but they have, they've evolved longer really than anything else on the planet. And they have vacuoles inside their unicellular little bodies that say when there's, less light, I'm gonna float to the surface, and that's where I'm gonna find the light. So that's one huge advantage. Another huge advantage is they're able to take nitrogen from the atmosphere. They're able to fix nitrogen. So our atmosphere contains a huge amount of N2 gas, and a lot of types of bacteria can harness that, and I won't get on that. I won't digress that way. But one advantage of cyanobacteria has is that they can grab nitrogen from the atmosphere. So sometimes when we see water conditions that have low amounts of nitrogen, high amounts of phosphorus, cyanobacteria can outcompete other types of phytoplankton. And then, of course, I'm sure not always, but at times, cyanobacteria blooms can create toxins that can be harmful to plant or to pets and animals like people. So I'm sure you know, but if you see a cyanobacteria bloom, you should not enter the water for that reason. We have a network of volunteer cyanobacteria monitors. Is anyone in this room a monitor? Okay, great. So that is a great partnership between Lake Champlain Basin Program, Lake Champlain Committee, Vermont Department of Health, and others to mobilize community scientists to monitor water quality visually. And then we have a protocol for folks to go out once a week and make an observation of water quality. And if they see cyanobacteria, they report it. If they see clear water and no cyanobacteria, they also report it. So we get all types of information. And this is all based on the evidence that was collected back in around like 2010, 2013 or so, where we took a lot of water quality samples and determined that if you cannot see cyanobacteria visually with the naked eye, then there is no evidence that cyanotoxins can be present. So that's really encouraging that we're able to use our data, use our analyses to make that kind of safety judgment. And then because of that, we can train volunteers to go out and use their eyes. It's much more data that we can gather by sending our teams of scientists out and driving all over the lake. It's just a lot more eyes out there. People like you who care about the lakes and care about water quality. Yeah.
And this cyanobacteria blooming in Lake Carmi, we're all familiar with that. When it's just green, can that be toxic also? Before it turns to aqua blue,
Right.
And all those colors.
It's possible. So if you can see a cyanobacteria bloom with the naked eye, and you can possibly identify it as cyanobacteria, so you're doing things like very carefully, but if you cannot pick it up with a stick, and you can do a jar test, take a glass jar, wait 10 minutes and see if it floats to the surface. There's a few things you can do to identify it. But if you can see it, then there is an elevated risk of there being toxins present. It does not necessarily have to be a surface scum or have a blue color.
So if we took a jar of the lake water and it doesn't float to the top, it's not cyanobacteria?
That's an indication that it might not be cyanobacteria.
Might.
Right. I think I don't want to get into all the identification. That's a whole other hour-long presentation to talk about how to identify cyanobacteria. So I can probably just direct you to some resources. There's a lot of good training videos and things like that.
The whole lake is green.
Right.
So we're obviously not gonna swim here, but if we go in our boat
Yeah.
and it's green, is it still safe?
If it's green, it's probably a cyanobacteria. If it's the water is universally green color, it's most likely cyanobacteria.
But is that continuous over the entire lake? So there are certain shores that get it worse. There are certain areas that, especially over the aerators, it's absolutely crystal clear at that moment. Can you swim out there? Like, there's a natural turnover, as we were talking about, when I was a kid, it was called purging. Like a natural turnover does bring green to the surface earlier in the year.
Right.
That seems safer to me, but I'm not a scientist. But once it gets hotter and then you start to see this kind of look, yes, we're not gonna go in. But I guess my question is, if it's on certain shores, but it's not on other shores, if it's clear with your aerators, grandchildren going in on the aerators. Is that safe?
Right.
I guess that's my question, because yes, there's green, it kind of, well, and it disappears at night. At 5 AM, when I get up in the morning, it's crystal clear, like,
Okay, yeah, thanks, all good questions. So maybe I'll start with the kind of geographic spread of it. So if it's on. So if you have a cyanobacteria that's really limited to a shore, and it's maybe 10 feet out, it's maybe I would call it minor bloom. It would be generally safe, I would say, to swim on the other side of the lake where there is no bloom. Okay, so it's not, it's not gonna spread by magic to the other side of the lake instantaneously. Conditions can change over time, and if it's setting up on one side, it's possible that's gonna set up on the other side, so just be aware of that, of course. But if you're in an area where it's visually clear for as far as you can see, and if there's a bloom on the other side of the lake, then that is, that's okay, you can still swim in that area. You're talking about early season, it having a greener color. I would go back to those resources I can share with you to kind of possibly identify cyanobacteria. Water can naturally have a relatively green color, and there are natural or cyanobacteria also natural, but there are non-cyanobacteria things that can make water green. There's green algae, there are plants, duckweed, so that can be not necessarily cyanobacteria, especially if it's at the time of the year you wouldn't expect to see it. If you live there for many years, you wouldn't expect to see it in May or early June, right? And then for the nighttime. So if there's a bloom in the morning and it seems to go away at night, I would probably advise to not enter the water. So for public beaches, the guidance is when they see a cyanobacteria bloom, they are collecting samples and monitoring their cyanotoxins, and they're waiting really for that cyanobacteria bloom for about 24 hours, to clear for about 24 hours. So I would say that would be a better approach.
And I find the tracker, and so like if it's, it's at the state park.
Yeah.
And we're right across from the state park.
Yeah.
Then when we go in, it didn't look that green, but now the state park is.
If you have eyes in the water and the water looks clear, I would not be concerned about a localized bloom on the other side of it.
I'm not sure what clear means. If we put our feet in, how far down do we be able to see for the clear that you're talking about?
Yeah, sorry. So this goes back to being able to identify cyanobacteria. So clear is, I shouldn't have said the word clear, that can be misleading. What I meant to say is if you look at the water, you cannot see cyanobacteria. So you're not seeing pinhead shape or size, bits of green. You're not seeing what looks like a paint spill of green scum on the water. Obviously, that'd be a giveaway. But if you're seeing little pinpricks of bright green or bright green and blue, then that would be cyanobacteria. If you're seeing just brown murky water, or maybe it's yellow pollen or other things, that is generally safe. That is not something that's going to be potentially toxic.
Well, people do the second disc thing, and I don't think it's ever clear. Maybe it is, but
yeah. So
I've
again,
never seen the bottom of the lake.
yeah. Again, I shouldn't have used the word clear. When I said clear, I meant no cyanobacteria.
Also, if you find it, the list, be sure to one resource to find that light site.
Yeah, there's a lot of great resources out there. Should I keep going? Got more science to talk about. We have a whole discussion period too. Yeah.
Can you say again, what makes the cyanobacteria come to the top?
Yep. So cyanobacteria. The question was, what makes cyanobacteria come to the top? So again, they don't have brains, but I can't help but thinking of them as having little brains. So they, physically, cyanobacteria cells can physically react to the amount of light they're receiving, and when they're not receiving light, they fill up. They generate gas underwater and fill up a vacuole, it's called, and float to the top. And that's because they need light to photosynthesize and create energy and reproduce. So they're reacting to the amount of light and floating to the top, whereas other things are not able to do that. Other phytoplankton in the water, not able to do that. And so that's why when the conditions are right for cyanobacteria to proliferate and bloom, they're able to all come to the surface and they're getting nitrogen from the atmosphere, they're growing because they have nutrients available, and they're out competing other things that are also growing in the water.
What are those quote right conditions?
Yeah, so conditions that favor, we say favor cyanobacteria blooms, warm water. Lots of nutrients, lots of phosphorus. Yeah, light during the late morning, typically they're setting up around 9:30, 10 is when you might see them really start to set up. Low wind speed, and we'll talk a little bit about that if I get to it. Low wind speed because it's not mixing, and it's when there's low wind speed, they're able to take full advantage of that their competitive advantage to be able to float better than anything else. If the wind's mixing them, then other things are able to compete for that sunlight, and other things are happening too. So low winds, high temperatures, lots of nutrients. Yeah.
And I understand about the mixing and that was fabulous. How much of that knowledge was in 2018? Did they know that when they decided the aeration would help our lake? Some of us, is it really helping it now?
Yeah, let me get to
Why did they pick that?
let me get to the next piece. I wasn't involved in the decision
I
making
know.
for aeration, but I do understand their motivation and their the primary goal of the aeration system is a good one. So let me get to the next topic. I think it'll maybe help with that.
Matt.
Yeah.
Think quickly, don't worry about the timing as far as like when you have a Q&A.
Okay.
This is a productive discussion going on.
Yeah, right. We're kind of doing it as we go.
Yeah.
It's great. Okay, cool. So I'll get into phosphorus specifically, and this I think will tie into the motivations for the aeration, the primary motivation. So phosphorus, we talked about how there's excess of phosphorus from things like farming and development, from wastewater, but naturally without humans around, the only source of phosphorus would be very slow weathering of rock, of bedrock and from soil. So we've really added a ton of phosphorus based on or compared to that. And of course, people figured out a long time ago that if you add phosphorus to crops, it greatly increases biomass and makes the crop more bountiful. And of course, the excess phosphorus can run off, into rivers, into lakes. We've talked about this already. So I touched on this, but here's a little diagram to show that how phosphorus is unique compared to other, critical things for a lake ecosystem. Carbon can go, between the lake and the atmosphere. If you it's hard to see the difference in color, but this is supposed to be the water surface in the atmosphere here above it. You can see kind of blue and white. Sorry for the contrast there. Carbon, carbon dioxide can go between the water and the atmosphere. Things can use it to photosynthesize. Animals and plants can give off carbon dioxide just like we breathe, and that fluxes between the water and the atmosphere. Nitrogen, we talked about how nitrogen gas entering the atmosphere can be taken up by cyanobacteria. But phosphorus does not live in the atmosphere. It does not have a volatile gaseous phase. It's not floating around us right now like nitrogen and carbon is. Okay, so that means that what's in the water column is what cyanobacteria has to use, and it sticks around for a long time. It cannot be, exported to the atmosphere like nitrogen or carbon can. In the Lake Champlain Basin, this is work done by someone who was in my grad school class, Michael Waronin. He looked at the balance of phosphorus in the Vermont portion of the Lake Champlain Basin since the kind of early 1900s. And since that time, there's been a surplus of phosphorus, import export of 1,000 to 4,000 metric tons per year. So this means there's hundreds of or tens of thousands of metric tons more phosphorus in the Lake Champlain Basin than there would be naturally. Right, so this shows why there's an imbalance. We're importing more than we're exporting as food products. And so anything that has kind of left over from this surplus is called legacy phosphorus. So legacy phosphorus is kind of here from before, from years past. Things that were imported as feed or as fertilizer, and now it's either in agricultural sediments It's on the surface of our streets, it's at the bottom of our lakes, all examples of legacy phosphorus. This is the phosphorus cycle. And I only put this on here because I borrowed this from another talk and my wife told me I'd be crazy to show this to anybody. And so I simplified it like this. You can thank my wife. So water and sediment. So I'm going to kind of use this model to explain a bit about legacy phosphorus and why the aeration system was the chosen avenue. So again, legacy phosphorus, it's hanging out in sediments at the bottom of Lake Carmi or in all of our lakes. And it can be released. And when it's released, it's something we call bioavailable phosphorus. It's dissolved and it's readily taken up by things like cyanobacteria and other phytoplankton, macrophytes and paraphyte and all those things we talked about. Can take it up, especially when it's released from sediments. And a lot of things kind of determine how and when phosphorus is released. But the key I want you to remember is that oxygen is key to when this phosphorus is released. Low oxygen at that boundary between water and sediment means it's more likely for the phosphorus to be released when it's low oxygen. And if you think back to especially dimictic lakes or polymictic lakes we talked about, those boundaries are those parts of the water column, parts of the lake are not mixing. So everything that's kind of living down at the bottom of the lake is using up all the oxygen that's there and it's not being replenished by the atmosphere. So when you have a stratified lake, even slightly stratified, that heavier, more dense water, there's going to be less oxygen in it over time because things are using it up. And when there's less oxygen in the water, that can lead to the conditions where this legacy phosphorus can be released. Okay. Now in shallow lakes and shallow areas of a more morphologically diverse lake like Lake Champlain that has deep and shallow areas, the shallow areas are where this is most important because essentially the bottom sediments are close to where sunlight can reach, right? So
What depth is shallow?
yeah.
In feet,
I mean it.
please.
Sure, I would say this can be 50, let's say 30 to 40 feet would be shallow.
Our whole lake.
So Lake Carmi I would describe as a shallow lake, yes. I'll come back to it, but good question. So shallow areas or shallow lakes, the legacy phosphorus is released into that photic zone. The photic zone is generally, you remember that second depth, right? About twice the second depth is as far as light can reach. So in Lake Carmi, it's not so much that the light is reaching to where the phosphorus is being released, but we talked about it mixing over time, and it's not very far for it to mix to then get to that photic zone. Okay.
Isn't the aeration system mixing the phosphorus and making it available?
So the aeration system, my understanding is that it is causing some mixing, but it's not like a full turnover mixing. It's essentially making the difference between the top layers and bottom layers less drastic, so that it can be more readily mixed by wind. And wind is the more important mixing driver of mixing for Lake Carmi or for base. It's just more energy from a windy day than from the little bubbles that are coming out from the aeration system. All right, and then the other side of this is that for deeper lakes, where that phosphorus release is, much further removed from where light is, so it's not going to be as impactful for deeper lakes, and that's why we generally don't have as much problems with cyanobacteria in the deeper parts of Lake Champlain versus the shallow parts, and why again Lake Carmi, generally not a very deep lake, is more impacted overall. The timing of this is also really important because you have, we talked about phosphorus and other nutrients coming in from the watershed, and that of course is delivering, things like we use for cyanobacteria growth, but you also have this legacy phosphorus. So in general, most of the phosphorus coming in from rivers is happening in the springtime, snow melt and spring rains. That's when most of the nutrients are coming in. But at this time, the conditions aren't as right for cyanobacteria growth. Back to your question, Al, the temperatures aren't as high, it's cooler waters. It's generally windier and kind of choppier in general. And, you know, other things, diatoms and other green algae are growing. It's not really the prime cyanobacteria growth season. But when later in the season, in the summer and the fall, you have generally more stratified conditions, lower oxygen, warmer water, other conditions that favor growth. That's when legacy phosphorus is coming out into the water column, and that's when cyanobacteria might be more likely to grow. So that's why this kind of release of bottom sediments is thought to be critical for lakes like Carmi and for parts of Lake Champlain that are more shallow. And unfortunately, climate change, not just our land use and management, also climate change is making this worse because we have longer, more persistent time, dry weather, warm temperatures, you know, droughts, and then rainstorms as well, and midsummer rainstorms sending nutrients in. If you think about, you know, fall turnover, that's happening, you know, in the midst of that can be happening in October, November, but then we can get a heat wave after that happens. And that in the past for Carmi has caused cyanobacteria disease. Again, going back to the aeration system, I think now might be a good time to get back to your question. What is your name?
Andrea.
Andrea. Back to Andrea's question, which was, you know, why did this state of Vermont choose to go with the aeration system? It goes back to how legacy phosphorus works. We know legacy phosphorus is critical, and the idea is to increase the amount of oxygen at that sediment water boundary so that it's not released. And then if it mixes, then it won't be there to begin with, right? Because the water will have been oxygen. Will end up a secondary factor. So in my understanding, based on our monitoring, is that worked. Oxygen was increased, and there's probably less legacy phosphorus released. So that primary goal was achieved, but it also had a secondary effect, which was to reduce the amount of energy needed to mix the lake. And so that, because of that, a lighter breeze, for example, caused the lake to mix more readily and brought the nutrients that were still being released, even if it's less so, to the surface where it's out of activity. Let's see what else we have. I have a few things about climate change. You know, I just talked about it for cyanobacteria blooms specifically, but you know, there's other impacts of climate change, more frequent persistent rainstorms, reduced ice cover later in the winter and earlier in the spring. Lakes are, you know, just reduced ice cover overall. This is a really simple graph for Lake Champlain. We have data going back to the early 1800s, but it's most reliable going back to 1906 when the National Weather Service started tracking it officially. These are years when the Lake Champlain surface either froze over completely in blue or did not freeze over completely in red. And you can just kind of visually see there's a lot more red on the right side, right, in later years. So this is just a huge impact for Lake Champlain and other lakes in the region. So, you know, back in the early 1900s, you can see, you know, for a generation, the lake froze over completely nearly every year. Today, we can expect the lake to freeze about once every 4 years. And if this trend continues by 2050, the lake might freeze just about once every decade. And so it's a profound difference, not just in the ecosystem, but also, you know, our experience of the lake, looking out and seeing liquid water rather than ice. This is true. We don't have, as far as I know, we don't have good data on ice cover in Carmi, unless someone knows, and I don't know who's been keeping track of this throughout the family for generations. Secret data sets, no? What was that? Ice out contest. Yeah, that's actually there have been a few studies of those. What's Carmi do? They did a pumpkin or
A clock.
a clock? It stops the clock. That's great. Okay. Yeah. Yeah. I know there's some towns do pumpkins and whenever they fall to the ice, and that's actually there's a study that we try to use all that type of data for ice out. It's hard to determine when ice in happens or when it freezes up in the winter. So thinking about winter and freezing, I also want to bring up chloride pollution or road salt. So this is another like really visual source of pollution. It's something that, it's a relatively recent idea that our roads will be clear of ice and snow within a day, within a few hours of the storm. That dates back to the 1950s or so. Now we have a cultural expectation that we can, drive very quickly in the winter. This has had a profound impact on fresh water quality throughout our region, throughout the world. And so road salt usually is sodium chloride, NaCl. It's the same compound that you have on the table, your salt shaker. And that, when it enters water, so it's a little rock that you see on the roadside or on the sidewalk. And when that enters the water, it dissolves into sodium and chloride. So sodium, that's the same thing that, maybe your doctor says you shouldn't have too much of in your food or whatever. And chloride, that is a natural, naturally found in all lakes, but it's being elevated kind of like phosphorus is. And I think I have a graph here. Yes, this is another one from Lake Champlain, but we're seeing, steady increase in chloride levels, since we started looking in 1990 and especially in the past decade. Chloride is, this is not approaching a danger for drinking water in most parts of the Lake Champlain Basin. We do have some groundwater wells that have been contaminated with chloride, but it is an ecosystem concern. So even like low levels of elevated chloride can be toxic to the bottom parts of our food chain, of our food web for the ecosystem. And then when you're reaching, so we're at between 10 and 20 micrograms per liter. And when that gets into the hundreds, then you have acute toxicity for fish and beer. So this is, something that we're working on to try to reduce the amount of chloride that goes downstream. I also wanted to touch on aquatic invasive species. So who's familiar with this type of aquatic invasives? Probably everyone. Okay, so I won't talk too much about it, but I just wanted to mention that this is a main challenge that we have in the Lake Champlain Basin. And add a reminder to always clean, drain, and dry your boats when you're moving from even one part of a lake to another part of a lake. Clean, drain, and dry to prevent the spread of aquatic invasive species. This graph we kind of redesigned here. It's hard to see for you all, but you can see it's kind of growing over time from back to the 1880s. And it's to illustrate that once an aquatic invasive is in a lake, it never goes away. These lines are never going to go down. Okay, so the top one is non-native species. These are things that are not native, but they're not necessarily causing harm yet. We haven't documented any harm. And invasive species are ones that we know cause harm to the ecosystem. And I just want to say a few things about how we monitor for water quality and how this works. So I talked about filling a bottle and bringing it to the lab, but when we're on a lake, this based on what we talked about, you now understand as a novice that the surface is not always the most important part of a lake, right? You want to know what's going on at the bottom, especially in the middle of that thermocline and at the surface. So we use something called a Carmi sampler that's on the right, and this is kind of like an open cylinder, and you can drop it down to whatever depth you want, and you send something called a messenger down a rope. And it slides on the rope and pops the thing closed, and then you can bring water from that depth up to your boat, put it in a bottle, and take that to the lab. So that's something that we use to sample throughout multiple depths. Sometimes we're think back to that lake stratification idea. When we want to know what's going on with the equilibrium, the top layer of the cake, we might sample at 6 feet. And then right in the middle of the equilibrium, and then 6 feet above that thermocline there. So that way we get kind of an integrated sample or a mixed sample of what's going on throughout that whole layer of the cake. And then we do the same thing for the hyperlinear, the bottom layer of the cake. We can see how they differ in oxygen concentration, pH, and all sorts of different things. And here's just the graph on the right is just total phosphorus for Lake Carmi from a couple of different monitoring efforts. And I'm not even gonna try, you probably can't see it in the back. So that's just an example of a graph we can make to learn about the water chemistry of Lake Carmi using these types of sampling methods. And has anyone seen the Lake Carmi platform? Probably all have, right? So this is another type of water quality monitoring. We have, you know, a floating buoy, and I say we, it's not me, it's the UVM, actually my graduate school advisor is leading this project, and, you know, in partnership with the state of Vermont. And they have, something called a water quality, monitoring sun. It's, I should have had a photo on here, but it's essentially, a big cylinder, and it's got these little probes on the end, and it shines light into the water and can measure different types of properties of water, chemistry, and light. It's really, really cool. And from that, we can measure water quality every, say, 15 minutes, rather than every week when we go out with the boat and collect a bottle. So, has anyone been to the platform day website? No one. Andrew?
We've seen a presentation.
Oh, okay, well, you gotta go to the website. This is from this morning, so you can see, you know, tying back to what we've talked about. These two rows here, surface dissolved oxygen, 11.23 micrograms per liter. Bottom dissolved oxygen, 7.38 milligrams per liter. So it's just a milligrams earlier. Differences in dissolved oxygen from the surface to the bottom. That's not too, it's not a concerning level. But you can see that there's a difference. So, as an example,
Temperature.
it is.
Surface.
It is.
Where is that website?
I don't have the URL off the top of my head. What I did to find it is I said Lake Champlain buoy, I think. It was the top result or so. Maybe for a second. So yeah.
Is the information about the amount of energy needed to mix the lake, so we're having this comes to the moon, was that new information gathered from this monitoring site?
That was an interpretation of the data that was collected with this buoy. Yes, and among other data.
Measures the wind speed also.
This one does, yeah.
They're up for the. We're talking about legacy phosphorus. We're talking about total phosphorus.
Yeah, sure.
Now,
Yeah.
what we got,
Yeah, sorry, I'm throwing terms all over the place here. So thanks for that question. So we say total phosphorus because phosphorus comes in multiple forms. Okay, and you remember, maybe you remember, I was talking about bioavailable phosphorus. That's what plants and phytoplankton can take up right away. We call it bio. You know, life can use it right away. Bioavailable. Total phosphorus we use to just mean all types of phosphorus. That includes bioavailable. It includes particulate phosphorus, which is stuck to sediments. It includes organic phosphorus, which is part of organic matter that's dissolved like leaves that might be broken up in the water. So all these different forms make up total phosphorus. And so I threw that out very quickly. Sorry I didn't explain that, but that's all that means. Yeah. And so legacy phosphorus is just describing where it came from, I guess, or just an idea that it can stick around. And then total phosphorus is more about how we measure it.
so. We have legacy phosphorus, which is basically out in the pyramid.
Yeah.
Then we have phosphorus that actually comes in the water from the river. All kinds of. And so. Are we able to determine what impact adding phosphorus coming in has on cyanobacteria activity versus legacy phosphorus?
Yeah, great question. So they both have an impact. So like repeating the question, can we tell the difference between, the maybe external sources of phosphorus coming in from rivers and watershed?
Saying is, we didn't have any, we had legacy phosphorus. And we had from the area. Yeah, with that legacy phosphorus be enough for us.
Right.
It's treated with.
Yeah. Yeah, so in this case, it's maybe you've heard, I always kind of relate it to climate change, and maybe you've heard that if we stop burning fossil fuels today, we'd still be dealing with the effects of climate change for generations, right? Unfortunately, it is analogous, similar for a lake like Lake Carmi or for parts of Lake Champlain. The legacy phosphorus issue, even if river inputs, stop today of phosphorus, we still have a legacy phosphorus issue to deal with. It would be likely, I would say, probably still continue to cause blooms. And that's, however, the legacy phosphorus is there because of watershed inputs, right? Because of the river inputs. So it's not a reason to give up on the watershed. And you all know probably that there's something called a TMDL for Lake Carmi. I think it's in one of my slides coming up, but that's the budget for how much phosphorus can move downstream to Lake Carmi. And my understanding is that in 2021, that was achieved. I think it was 611 kilograms per year reduction. We're now meeting that target, but 1 year being that target is not going to make a difference. It has to be many, many years of reducing those watershed inputs. So, both can contribute to cyanobacteria. The little bacterial cell doesn't care where the bioavailable phosphorus came from, whether it came from a river, from legacy, from lake ponds sediment. But yeah, I think that on a seasonal basis, depending on the time of year, what's going on in the lake, legacy phosphorus can be a more important factor, or river inputs can be a more important factor. Yes. All right, I think I had a few more slides. So I just wanted to mention other types of water quality monitoring going on. We have similar types of buoys in Lake Champlain and in the Lamoille River going into Mass Bay. And there's another website you may be interested in. This is data.lcbp.org, which has data from around the Lake Champlain Basin. It includes water temperature for Lake Champlain and water quality parameters for Lake Champlain. The temperature graph of near Valcour Island came from this website, and you can also see a discharge way back. It was an hour ago. We were talking about how much water moves downstream in a river. All the discharge data from all over Lake Champlain Basin goes to this website. So if you want to continue your journey as a limnologist, you can check out this website. And I also want to just talk about how the Lake Champlain Basin works a little bit. We use what's called an adaptive management model. So we have, of course, pressures on the landscape and on our lake due to pollution and land use and human activities. And we use science to determine the state of a lake through monitoring that we talked about. And then we develop a response. And we have, a management plan. And for the analogy for Lake Carmi would be the Lake Carmi action plan. And then we kind of do it all again and continuously adapt to the pressures and the state of the ecosystem. And so we have 4 main goals: clean water, healthy systems, thriving communities, and informed and involved public. And we report on those goals and assess them in our state of the lake report, which we put out every 3 years. The next one's coming out in 2024. So keep your eyes peeled for that. I also, if you're here, I thought you might be interested in our science blog, which is not written for the general public, but I don't consider you all the general public because you've all learned a lot today and you clearly are interested in this stuff. So this is really written for watershed managers and people who are doing this work. And it's really about the latest research we have on water quality in the Lake Champlain Basin. A few staff members and I write these. We don't start with the basics. We start with what are the findings of this research and how can it be applied. And we're basically taking a 100-page technical research report and distilling it into a few minutes of reading. So you might be interested in this this science blog. And then I just want to, we've already touched on a lot of this, but I wanted to just bring it around to Lake Carmi because I know that's why a lot of you are here and a lake you deeply care about. So we talked a bit about the phosphorus TMDL. That's being achieved, but it needs to continue to meet water quality goals. Monitoring continues through UBM and state of Vermont, and partially the Lake Champlain Basin Program as a partner as well. We talked about the aeration system. I believe this is the last year it's I'm not sure what the latest is. This is planned to be the last, or they're going to do it again next year. No, yeah, okay. I know that discussions about alum treatments are in the works, that aluminum sulfate. Similar idea to aeration, but it's a different, similar goal, but really different mechanism. Okay, so aeration we talked about, the goal was to increase the amount of oxygen, right, at that sediment water boundary to reduce the release of that phosphorus. Aluminum sulfate is applied to the water and it binds up with phosphorus and then precipitates out. So it doesn't matter, it doesn't care if it's legacy or if it's river inputs. It kind of just chemically binds with dissolved phosphorus and then falls out of the water column. So there is less phosphorus available for phytoplankton, cyanobacteria to grow. Okay, this is established chemistry, we know it works. It's only appropriate in certain lakes with certain, water quality conditions and pHs, and a lot of studies needs to be done to see if it's appropriate. Place to use alum, but it has, we know that it takes phosphorus out of the water. That's not the question.
Say that last sentence again.
Yes, I was just saying that with alum treatments, we know that alum will bind with phosphorus and the phosphorus will fall out of the water. The question is whether it's an appropriate lake to use alum and whether it will be effective and what time frame it will be effective. And there's a lot of unassociated questions to answer, but the chemistry is well established. Yeah, do you think that's a good plan for this lake? Great question. I'm not going to answer that today. As a scientist, I support extensive research and planning, and I would need to see all the studies. Okay. Yeah.
Can I ask a related question, Chairman, which is on aluminum sulfate alum? Do you think, as a scientist, that science knows what needs to be measured and understood about Lake Carmi to determine alum is or is not a viable course of action? Or there's some unknown?
Yeah, that's a great question. I would say, I think we do understand it well enough. I think we can, we understand the science well enough to determine whether it's an appropriate action. And, it's not a cheap course of action either. So really, there's a cost benefit analysis as well. And when you're doing watershed work, you're investing in the long term. And when you're doing alum treatments or aeration system, you're spending a lot of money in the shorter, for a shorter term solution, while a longer term one takes effect. But this, alum has been used, it's used more frequently throughout the country and throughout the world in drinking water reservoirs and smaller water bodies. Lake Carmi, I think, would fall into a category of lake it's been done many times over throughout the country. And it's been monitored for decades, and I think we have a good understanding of how it could be applied to Lake Carmi if the cost benefit, relationship works.
I'm sorry.
If they don't go with the alum treatment, they have aeration, that's sort of a mixed bag. Is there anything else for us to think about doing? So our grandchildren can go with it.
So the options, there's only, as far as in-lake treatment options, there's only a few categories. There's dredging, which means you literally go in and pull, dig sediment out to physically remove the lake's phosphorus. There's circulation, so you can just move water around and mix it, which, of course, we know that's not always a good idea to mix water. There is aeration, the bubbling, which was done on Lake Carmi. There is, and there's chemical treatments, including alum or other chemicals that precipitate phosphorus out. Those are the categories of inlet treatments that have enough of a track record that would have any applicability for a water body. So that's why the best course of action is to look at feasible in-lake short-term options while aggressively pursuing watershed solutions and reduce loading. The problem with nutrient loading Lake Carmi is not a new one. It's been phosphorus and nitrogen pollution to Lake Carmi has been going on for over a century. So we've only been working on a solution for a handful of years, in comparison. So it's not fast. It's not fast. So that's not the reality. And again, it's not unique to Lake Carmi, unfortunately. It's kind of happening over the tree on the world.
How treatments only last for a short period of time?
Yeah, it depends on, how much Allen's applied, and that's part of the study is, there's a lot of different ways to do it. You can do a little bit at a time, kind of continuously, or you can do like a major, dosing for 1 year, and it might last a handful of years. So that's part of the trade-off, the cost benefit.
It's expensive. Good.
Yeah, there's going to be more expensive and it's very unlikely to happen. I said that as a category of things that can possibly be done. But that drugging would be more like artificial water bodies, managed for a specific purpose that have a severe contamination might be very. The Western US doesn't really happen around here for natural things.
You mentioned you go to inmate options, there's water that often, but I guess that's, two number invoices for the Carmi. And how does it ever get released? And the offers ever get released?
From the comment?
Yeah.
Yeah, right. It goes downstream. Yeah, there's only. Well, remember, Lake Carmi, back to my second slide, is at the bottom of something in the middle of something else.
Yeah.
Right, so Lake Carmi is part of the Pike River watershed, which goes into Quebec. There's no release of phosphorus to the atmosphere. But phosphorus, I can show you the crazy graph, all the arrows again as well, but it's changing forms and being processed in all different ways all the time. So it's not just sitting there, and it can get buried too. So it can kind of it's moving throughout the lake, throughout sediment, boundary there, and it eventually is moving downstream. And we're thinking on geological time scales. We're in a blink of an eye right now. For human time scales, it matters in eternity.
You mentioned cost benefit ratios quite a few times. I understand that you have cost aspect of it, but when that cost benefit ratio is done, what includes better? I mean, how do you measure that? Obviously, it would be wonderful to have people swimmable fishing with water. But what is taken? You may not get the answer to kind of learning what you think. I'm just curious from the benefit side. Okay, it's going to cost $400 to put it out for them. What's the benefit?
Right.
And how do you quantify?
Yeah, I think I may let your answer be as rhetorical on that.
Yes.
Yeah, but it's a great question. And you all know that the state is really taking the lead on evaluating options and, I think they targeted, the primary outcome of their aeration system. I think they reacted and responded to the science really well. And, I know that they're working with your community. When they're looking at the alum, these are people who also care deeply and are trying to do their best work. And we have, when I describe my job, I say that it's great for a lot of reasons, but especially because we have shared goals. Everyone wants the same thing, right? We all understand what it would mean to people to have a bloom-free lake or seasonal lake online. That is clearly, there aren't folks working in the state aren't robots who don't understand that, right? So in terms of quantifying it, it's difficult to quantify a benefit like that, but clearly it's incredibly valuable to have water that you can enjoy. That's my attempt to keep it virtual. Yeah.
Just to follow up on the previous question, I know that there's a no-coil harvester in the lake. That's effectively using phosphorus in the lake, as long as it's being moved outside of the lake on water. That small, it's like threatening plants.
Yep.
Does anyone know where the no-coil is? On the coast. Yeah.
Right. So you're not.
So it's not jumped out of the watershed?
Yeah, it's, I thought it was, it's not out of the water.
Right. Out of the lake.
Still in the water.
Exactly.
Yeah, just depends a little bit back.
Right.
Over here. Right.
Can I ask you a question?
Sure.
You talked about 4 main goals, including clean water and thriving communities, et cetera. With the exception of cost, which is obviously a major factor, are there organizations or other interests that would oppose treatment of the lake to achieve those goals? That is other than natural.
Are there so I mean,
Who doesn't like the idea of making the lake swimmable and
what groups might oppose alum treatment is the question?
that and other community wants a clean lake, who might not?
No organizations come to mind who would, specifically oppose that action.
Is it then?
But again, that's I maybe think about the idea of the action. We'd have to see, all the specifics of a project.
Would it be fair to say that if not the primary reason, a primary reason is the cost for implementing a solution?
Primary reason for what do you mean?
For implementing a solution is the cost of it, the financial cost, and the budget from the state.
Right, that would certainly be a major consideration, so that would be expensive.
Are there any negative environmental impacts?
Yeah, good question. That, if done, in an appropriate way in the lake where it's suitable, there should not be major impacts. It does alum. One reason there is some potential concern is aluminum sulfate obviously means aluminum. Aluminum is a metal that is toxic, right? Only in certain water quality conditions. So essentially, pH of the lake is a major consideration for the appropriateness of alum treatment. So if the pH is outside of a range, may be appropriate or could become outside of a range or will be appropriate for alum treatment, then you would not want to move forward. That's the
We have more slides.
Let me see. I think I might have, that's basically it. I had some resources and I just pulled this up to talk about a few things on Lake Champlain, but I can turn it off if you want.
Or you can speak to like this larger geologic time frame of the evolution of lakes. If you're prepared, and that's where you can do that.
I can try. I thought about it a while, but yes. Yeah, so we have something called a term called trophic state of a lake, or it applies to each general, but the trophic state of a lake is basically how many nutrients and living things are there in a lake. Okay, so it can span from the first one, kind of like in the evolution of a lake or a life cycle of a lake. You start as a oligotrophic lake, which means essentially a star in the landscape, maybe a glacier carved a big hole in the ground and it melted and there's water there, but not much else. That might be an oligotrophic lake. So think about Cascades in Washington, Outbound Mountain, a cirque, beautiful landscape you might see in a magazine. That would be oligotrophic because it's a new, geologically new lake, mostly just water. There might be a few things, some bacteria and algae living in there, but fish, macrophytes, little paraphyton, not a lot of nutrients, oligotrophic. Then there's the next one would be mesotrophic. I've been talking for a while. Metatropic, right, so mesotrophic would be the next phase, and that's like between, kind of halfway in between. So that's an older lake, more and naturally, lakes, begin because their watersheds are sending sediment and nutrients to the lake, it becomes a more hospitable place for things to grow. So you have plants growing and then, things make their way, like maybe got stream fish start to come, hang out, you have birds start to, become part of the ecosystem, there's fish there and they're moving in the lake and all things are, the things are happening over thousands of years. And then the last stage of the lake is eutrophic lake. That is the highest level of nutrients. The lake is generally becoming shallower, sedimentation, sediments being delivered to the lake. There's higher nutrients, there's more primary production, and eventually, geologically, the lake would fill in, seems to become a lake, it might become a river or a wetland or whatever else, depending on the landscape. So, as natural, humans have essentially accelerated that time frame in strange ways. So we go from, an earlier trophic state to a much later one, in the course of 100 years rather than 10,000 years.
Is there any danger in eating the fish we're catching out of Lake Carmi?
I will refer you to the state's consumption advisory guidelines for fish, which I don't think is specific to Lake Carmi. I think it's statewide and Lake Carmi is applied to Lake Carmi. I don't know if there's any exceptions to that.
Well, we don't have real internet, do you sort of know what it is? Is it once a week, twice a week?
It depends on the species of fish. So that we'll have like a chart with pictures. And it says, if you got this, and it's split up by healthy adult and children and two categories of people who might be at higher risk. And they'll have guidelines for, I think it's monthly. I think it's like number of certain it's been lost, but basically they have maybe I think it's 8 ounce. I probably shouldn't say exactly, so I don't remember, but a meal is a certain number of ounces, and you can eat so many meals per month depending on the species and your risk level.
And where would we find it?
Yeah, that's fine. I would search for Vermont State Fish Consumption Advisory.
We can find it and send it.
Yeah, that's
Oh, that would be fun.
yeah. Yeah, and those are Department of Health. Department of Health, yeah, and Vermont Fish and Wildlife, those should be able to link to it. And those are all the Vermont fish consumption advisory guidelines are because of mercury in fish tissue. And this is another, we didn't get into this, but mercury pollution, is a result of coal fire power plants, mostly in the Midwest, that blows over here, blew over here in the past mostly, and causing our landscape onto our watershed near Swing Lake and Blue Ridge. Legacy, yes, go ahead.
So Lake Carmi is a natural lake, but it is also to be required in this year. From when you have any thoughts on the higher water level, will that have any effects on all of this good stuff?
Years.
Yeah.
You're welcome. So sorry.
I probably won't wade into this.
You want to comment?
No.
Yeah. I'm being recorded.
Yeah, I know. Yeah, I'll let that one lie, but yeah, that is true. I mean, maybe you're all analogists now, so you can think about things that might be different with the, different depth.
Well, I did notice because of all the rain, that it's rated the season that we had, the water didn't turn green. It's much later because there was so much, the water is probably, but still 6 inches higher than it usually is. The more rain does affect cyanobacteria, as I'm understanding, the movement, the mixture.
Yep. So I think that as it's a factor, like there are many factors. It's always, we get a lot, basically whenever anything bad happens, unfortunately, the media, the press is calling us up and asking us, well, this happened, so is that causing that? And we really, I look back to factors, is my favorite word for this, because yes, it will certainly be a factor. Temperature, when it's raining, it's also cloudy, cool, and windy. And so those are all factors, right? If the lake might be deeper, that's gonna be a factor. It might be more turbid, you know, back what Andrew was talking about, you can't even see your feet. That turbidity, we call it turbidity, is the measure of water clarity. If there's increased turbidity after a big storm, that, yes, there may be nutrients coming in, but also it's gonna limit the amount of light available for cyanobacteria growth. So it's a factor. Grill me anymore? That's.
Okay,
Like, for back, if